This is the video animated version of the Radwa and Radwa podcast, where we aim to communicate scientific research to science enthusiasts. We post 4 versions of each episode, in English and Arabic, and in podcast and YouTube video formats. All the links are in the description below. You will also find our references in the description. We are your hosts, Radwa Ewaisha
and Radwa Sharaf.
PhDs, pharmacists, and molecular biologists. In our episode today, we will be discussing the Human Genome Project. In our last 3 episodes, Radwa talked to us about DNA, RNA, and RNA vaccines. The link to these episodes is in the description below. Today, Radwa, could you please tell us a little bit more about the word genome and what it means?
Absolutely. So the word genome really represents the string of letters that are encoded in the DNA of one of the organisms that scientists are studying. And like the human genome, there is the bacterial genome, the yeast genome. So for every organism, of course, the string of letters encoded in the DNA will be different. And nowadays, the study of the genome is actually an entirely new field. It's called genomics, which is the study of the string of letters encoded in this DNA. And there are specific institutes and centers that are dedicated to the study of the genome. As we've already discussed in the episodes prior, our genome is composed of DNA, and that is consisting of 4 unique letters, A, C, G, and T, and these represent the letters present in our DNA.
So, how did the idea of a human genome project start? And do we now actually have the sequence of the human genome?
Yeah, so the idea of generating a human genome sequence has been around for a while, ever since the 1980s. But scientists haven't actually started on the project until 1990, and it took them about 13 years to complete the project. It was a collaboration across 20 separate universities and research centers in 6 different countries, where scientists worked on reading the letters or sequencing the DNA of human volunteers. These volunteers were recruited through an advertisement that was released in the New York newspaper in 1997, asking volunteers to come in and provide blood samples, and these samples were used to extract DNA in order to sequence the human genome. Over time, as scientists were working on these samples, some of them were somewhat easier to work with, and they had more success in amplifying libraries from one specific donor. And so today, the human reference genome consists of about 70% of that genome is originating from one specific donor for that reason. And the whole process really took quite a bit of time. So, using the technology that was available at the time, also known as Sanger sequencing, it took scientists about 6 continuous years of sequencing 1,000 bases per second, 24 hours a day, 7 days a week, to generate the human reference genome at an estimated total cost of $1 to $3 billion. Of course, the actual cost is a little bit hard to pin down because the work was done across different countries, but that's the estimate so far.
Wow, 13 years and 1 to 3 billion US dollars. So, were they successful in the end? Do we now have the reference sequence?
It's an interesting question because over the years, scientists kept saying, we have the reference genome, only for us to find out that no, it wasn't really entirely complete. So let's walk you through the history. In 2000, President Clinton came out in a press conference with 2 of the most prominent scientists working on the Human Genome Project, and they said that now, at that time in 2000, they had a draft human reference genome. At the time, the genome was only 90% complete. That meant that 10% of the human genome was still missing, either because it was difficult to sequence using the technology at the time or it wasn't completed yet. And then 3 years later, in 2003, scientists published a paper showing that 92% of the human genome had been completed, and that reference was being used for scientific research at the time. But the remaining 8%, that 8% consisted of some repetitive sequence that was quite difficult to interrogate using the technology available at the time. And so over the years, the sequencing technology has improved. We've moved on from the old Sanger sequencing to next generation sequencing, and then later, even more developed third generation sequencing modalities that allow us to identify the sequence of these really hard to sequence regions that consist of repetitive elements. And only last year in 2022 were scientists able to announce that the entirety of the human genome, 100% of the human genome, has been sequenced. This is also referred to as the telomere to telomere sequence. Telomeres represent the ends of the human chromosomes. And so telomere to telomere literally means end to end. So now we can finally say that the entirety of the human genome sequence is known, and the reference is available online for anyone who's interested in looking at.
Wow. So, what did scientists discover, and how did those discoveries help us?
So, scientists made quite a few discoveries from the original Human Genome Project, but a human genome reference sequence was used across many, many projects, and so it helped inform a lot of research that was built on the reference sequence. Let's start with a few key findings. The first finding is that the human reference genome consists of 3 billion letters. Initially, scientists thought that the majority of these letters encode instructions for the synthesis of proteins that are used to inside the cell to actually elicit a function. But actually, they discovered that only a small proportion, about 1%, of the human genome encodes for proteins, which was quite a surprise. Scientists had hypothesized that a much larger proportion encoded for proteins. And initially, scientists thought that the 99% remaining DNA was junk. They specifically refer to it as junk DNA. But over the years, we've discovered that this is not really junk DNA. It still does encode a function, just that it doesn't encode for a protein, but it has regulatory functions that help decide when certain proteins get synthesized and what levels they will get synthesized at, and so on and so forth. So that was one key finding. The second key finding is that humans synthesize about 22,000 proteins. This number is much smaller than what scientists had originally hypothesized. We thought the number was closer to 100,000, but actually only 22,000 proteins are needed to elicit all the different types of functions that a human cell needs to do. And the third finding, and that only came about once we started sequencing more human individuals, that is, across the human population, our DNA is 99.9% identical. And all the differences that we see within humans only originate from differences in 0.1% of our human DNA. Now, 0.1% seems like a very small number, but remember, it's 0.1% of 3 billion letters. So 0.1% is actually 3 million letters, and that's still a whole lot of diversity. And so scientists have realized that it's crucial for us to not only sequence one or two or three reference genomes, but it's important to have many references that represent people across different ancestries and different backgrounds that help us capture all of the diversity or much of the diversity present in the human population. And now we have references that we can compare any human DNA that gets sequenced against. So someone might ask, why is this important? Well, this is very important because if someone comes in to a clinic and they have a genetic disorder, we can sequence their DNA, but if we don't have a reference to compare that DNA to, what use is it really to sequence their DNA? And so having that reference genome is crucial because now we can compare any individual's DNA against this reference and check for the locations where the human's DNA differs from the reference. And then we can try to do research to identify which of these differences truly did result in the disease that we're studying versus differences that are just part of the diversity seen across humans.
Thanks so much, Radwa. So, to summarize, the human genome project is a project that took so many years to complete, and it aimed to help us identify the letters in the human genome or our human DNA. We found that the number of letters is 3 billion, and that 99.9% of that DNA is identical between human beings, and that only 1% of that DNA actually encodes proteins. Now we have a reference genome to help advance both scientific research and clinical practice.
We hope you enjoyed today's episode. Please don't forget to like and subscribe to our podcast and our YouTube channel. You can also find us on Twitter, Instagram, and Facebook. Feel free to reach out with suggestions for future episodes or leave us a comment directly on our YouTube video. See you next time!